This video presents information about the mechanisms by which leaf greenness relates to water stress tolerance in potato. It is based on a research paper named Leaf Greenness as a Drought Tolerance Related Trait in Potato, Solanum tuberosum, and published in Environmental and Experimental Botany. The first step, before commenting the research work, is to define some important concepts that are going to be used along the video. The first one is trait. According to the well-known naturalist Charles Darwin, who introduced the concept, a trait is a prediction or proxy of an organism's performance in nature. A more modern definition, according to Boyle et al., is a trait is a measurable characteristic at the individual level. It may be a morphological characteristic, like for instance the pinnation of the potato leaf. It can also be a physiological characteristic, like a photosynthesis rate of a certain individual, or finally, it can be a phenological characteristic, like for example the moment when emergence occurs. The next important concept to be defined is leaf greenness. Leaf greenness is a general term that characterizes the color of the leaf in reference to the intensity of the green color associated to the chlorophyll concentration. It is highly correlated with the SPAT measurements. To assess this correlation, we extracted A and B chlorophyll with acetone from leaf tissue and measured its concentration by Lichtenthals and Wellborn method. They presented a formula that relates absorbance measurements by spectrophotometry at two red wavelengths, 646 and 663 nanometers. These chlorophyll concentration values were correlated with SPAT values from the same leaves. SPAT is a non-destructive measurement. It measures leaf absorbance difference between 650 and 940 nanometers. There is no leaf absorbance at 940 nanometers, so the SPAT measurement uses these values as a benchmark to estimate chlorophyll concentration by absorbance at 650 nanometers. The correlation between chlorophyll concentration and SPAT measurement has a high Pearson correlation coefficient of 0.78. The objectives of the study were to 1. Compare and relate the temporal patterns of physiological measurements across watering treatments and 3 different varieties, and 2. To relate water stress tolerance with different temporal attributes of chlorophyll concentration. The experiment was carried out at the International Potato Center Experimental Station under greenhouse conditions in Lima, Peru. 3 genotypes were tested: Cernaf, a recent material that shows high yield under water limiting conditions; Desiree, which has a moderate tolerance to non-severe water stress; and Unica, a genotype that has a good response to warm and dry environments. 2 watering treatments were imposed since tuber initiation onset. 1 control, in which the substrate was maintained at field capacity, and a water restriction in which the substrate was fixed in a gravimetric humidity of 50% of field capacity. 6 sequential harvests were carried out. The following physiological responses were measured in the present work. 1. Chlorophyll concentration with a SPAT meter, gas exchange measurements, canopy cover, leaf length, and biomass accumulation. After measuring all these variables, we found the following results. In this first figure, we found the temporal patterns of maximum net photosynthesis, chlorophyll concentration, and leaf length. The black bullets represent water-restricted plants, and the white ones, the control plants. The first thing important to notice is that water-restricted plants in the three genotypes presented higher chlorophyll concentration values than their control in the first 4 weeks after the initiation of watering treatments, but presented lower values for maximum net photosynthesis and leaf length. Chlorophyll concentration peak after water restriction was a consequence of leaves stopping growth. Leaf growth is known to be driven by cell growth and cell division, being turgor pressure the driving force for cell growth. In the last years, it has also been evidenced that root signaling might be an important factor for stopping leaf growth under very early stages of water stress. It is also important to note that Sarnaf, the genotype that presented the lower chlorophyll concentration peak after water restriction, is also the genotype that diminished less its growth. Remember that Sarnaf was a genotype with less differences in chlorophyll concentration between watering treatments, and as seen in this figure, it's also the genotype with the higher tuber and above ground biomass under water restriction. In this figure, DSI is presented in the y-axis. DSI is a drought susceptibility index. At higher DSI values, more susceptible is a certain genotype to drought and therefore to water stress. The maximum chlorophyll concentration amplitude between the control and water restricted means is presented in the x-axis. This plot suggests that not increasing the chlorophyll concentration under water restriction is a trait related with water stress tolerance. Another important result from this study is that the genotypes with longer development cycles, like Sarnaf and Unica, when compared to Desiree, have a higher final biomass accumulation because they have a longer period of time for transforming light energy into plant biomass. This is evidenced by the day in which senescence onset occurred in each genotype, as well as a temporal slope of chlorophyll concentration. Desiree water-restricted plants presented a more negative temporal chlorophyll concentration slope, which represents a faster senescence than the ones seen in Unica and Sarnaf. The conclusions of the study is that chlorophyll concentration measured by a SPAT meter is a trait related to water stress tolerance by two mechanisms. The first one is that the SPAT amplitude between watering treatments is related with the degree at which the plant stops growth. A low or no amplitude reflects growth maintenance, which under moderate water stress is the best strategy to cope with drought. 2. The second one is that delaying the senescence onset and having a lower senescence rate is directly related with the time that the crop has to accumulate biomass through photosynthesis and to translocate photostimulates to the harvestable organ. This research was conducted under the CGIAR research programs on water, land and ecosystems, root, tuber and bananas, and climate change, agriculture and food security. This study was part of the BMC-GIC bilateral project, Improved Potato Genotypes and Water Management Technologies to enhance water use efficiency, resilience, cost-effectiveness, and productivity of smallholder farmers in stress-prone Central Asian environments.